As scientists and researchers, we stand together in strong support of Assembly Bill 305 and Senate Bill 260 in the Wisconsin legislature that would restrict the use of abortion-derived fetal tissue for research. We do not agree that research using human fetal or embryonic tissue from abortions for procedures such as IVF is ethical or a requisite approach for advancing scientific inquiry or preventing suffering. The argument that fetal-derived tissues must be used in research to develop medical treatments is false. Many therapies have been developed using cell lines not of fetal origin, including insulin for diabetes, produced in bacteria, Herceptin for breast cancer, and tissue plasminogen activator for heart attack, stroke, and pulmonary embolism, both developed in Chinese hamster ovary cells. Other successes include 5 new FDA-approved drugs as of 2011, developed using the chemical glutamine synthetase system, and more than 70 successful treatments developed using adult stem cell sources. Even though the often cited polio vaccine was developed using fetal tissue cells, the developers of the vaccine later testified that initial studies were also successful using cells that were not of fetal origin. Therefore, it is misleading to suggest that important medical advances would not have been possible without using cells of fetal origin. We respect Dr. Raymond and our colleagues at the Medical College of Wisconsin and do not question their dedication to science and humankind, nor are we advocating for their criminalization. However, each one of their examples involves the hope that fetal tissues will provide life-saving treatments. Their argument substantiates our stance that no current treatments or therapies exist that necessitates the use of fetal tissues in research. To the claim that restricting the use of abortion-derived fetal tissue will cause research to come to a halt and an exodus of research talent from the state, we answer that we have experienced the opposite. Students and researchers have left science altogether after failing to find research laboratories that did not use abortion-derived or human embryonic tissues. Thus, the continued pursuit of these unethical avenues of research may cause us to lose brilliant minds, research grants, and possibly the talent needed to discover cures to deadly diseases. Ultimately, what matters most is that we cannot support the exploitation of one group of human beings, the preborn, for the benefit of another group. We became scientists and physicians to serve humanity and to study the natural world in order to improve the human condition. Compromising these ethical standards undermines our work and taints future discoveries. Nothing can diminish the fact that using human embryos or fetuses as objects or means of experimentation constitutes an assault against their dignity as human beings who have a right to the same respect owed to every person regardless of developmental stage.